my name is Natalia,
And I'm Abigail.
and we will be presenting on chronic intermittent hypoxia and how it disrupts/disturbs the regulation of breathing and drinking in awake and alert. Oh, I have to talk closer. Okay, alert mice. And we were working in the Ramirez lab. Yeah. So what we know is that, hello? Oh, okay, cool. Okay, so what we know is that 25 to 45% of pediatric cases deal with dysphagia, which is a swallowing disorder that affects breathing. And that, oh Lord, okay, what is this? And that 3% of pediatric cases involve obstructive sleep apnea. And of these are comorbid to other disorders. Where is this going? Okay. To other disorders such as congenital heart disease, neuromuscular/neurodegenerative disease, craniofacial abnormalities, and obesity. It is important to note that these are also prevalent in adults. In fact, they're the #1 leading cause of death in neurodegenerative disorders.
Chronic intermittent hypoxia, or CIH, is a widely used model that mimics obstructive sleep apnea. Obstructive sleep apnea, or OSA, is the obstruction of the upper airway during sleep. The closure of the airway occurs during intermittent pharyngeal collapse. And on the left, you can see a diagram where it details where in the pharynx this collapse occurs. To the left of this, the right of the schematic, is an anatomy diagram. And here we want to point out that it shows the upper airway as well as the lower airway. We also want to note that OSA is a human disease. As mentioned, CIH is a model for OSA, and the inspiration for our project actually came from an article that was published by one of our mentors, Dr. Huff. And this article looks at the interaction in swallowing, breathing coordination. What we currently know is that dysphagia, or disordered swallowing, can lead to aspiration pneumonia by not being able to coordinate swallowing and breathing. Here in this diagram, aspiration pneumonia occurs when food or liquids travel through the trachea to the lungs rather than traveling through the esophagus during normal swallowing. Aspiration pneumonia is also a leading cause in death in breathing and neurological disorders. What we currently don't know is if licking, drinking coordination with breathing is disrupted in OSA. And this leads us to our hypothesis that chronic intermittent hypoxia disrupts coordination between licking and breathing.
And then this leads us to our chronic intermittent hypoxia protocol, which we used in 2 chambers. The first being what you see in red on the diagram to the right, where it's cycled between 5 minutes of 21% oxygen and then 60 seconds of 5% oxygen at about 80 times per day for a total of 23 days, starting at 9 in the morning and ending at 5 p.m. in the evening. And then the control chamber just remained at 21% oxygen for all 23 days. To ensure the health and safety of the mice, we did weigh them daily, and their cages were changed weekly. We also used adult mice at P40, meaning postnatal. And then for our experimental timeline, all the purple regions are the time that they spent in the chamber. The green portions are the water deprivation periods. I would like to note that they were water deprived in order to have them get used to licking in terms of the chamber that they were put into. As a reward system, they would get sugar water. However, they did receive a daily set amount of water every single day during that time period. And then in the blue region is the free water they received during the weekend, and the yellow and red are the testing period days. And for those of you curious about the data, we used a total of 15 mice on a black 6 background, and then details on sex distribution are in the table to the bottom left. And then we used, oh, that was a little wonky, but it's okay. We used this setup as our lickometer and to acquire plethysmography. So that blue stuff that you see at the back behind that clear chamber is what records our plethysmography, which is highly sensitive. And where the lick spout is what records when they lick. So that's their lick sensor, and that's traveling towards that green box over there. And I'll show you what that reading looks like in a second so that you understand our data a bit better. And then what we did here was run 2 trials of 3 minutes each. We didn't do 5, even though we originally were going to, because we found that after 3 minutes, they were satiated and no longer wanted to drink. And when they when they stopped, they would either move around, jump, or do other behaviors that, because of its sensitivity, would kind of skew our data. So keep that in mind. And then as a note, the knitted mouse is not to scale. They are much smaller in real life. And this is a plethysmography example. Just for simplicity, we'll use the word pleth. And here it's a very zoomed in version. We'll zoom out a bit more when we go to our data. But just so this is for your sake to make it easier to understand. The x-axis is time and the y-axis is the change in pressure in terms of the sensitivity of the chamber. Upwards motion is inspiration and downwards is expiration. And then something to keep in mind is that this has never been done before. So keep that in mind as we go through our data. And then, as I said, we measured lick and non-lick bout durations. The orange portions in the lick sensor is what we measured. The blue ones are as lick, and then the orange ones are as non-lick. And it corresponds to the readings in pleth as blue and orange. And we use the root mean square amplitude. And you can kind of already see that there is a difference going on. In blue, the lower change in pressure implies that it's shallower and shorter breathing. And just as a reference, we used the percent max of baseline of the non-lick in order to compare our data. However, because of that added noise and external factors, some of the readings could have been skewed. So take that into account.
And here we have a pleth reading from our control group. We want to note that we didn't see a difference in the pleth reading, the breathing in the mice from comparing non-licking to licking. And we noted that this was reflected in our data as well. There is no significant change in this group comparing licking to non-licking, and there's also the difference in the in breathing in these mice and the root mean square amplitude as well. Here we have another pleth chamber reading. This one is from our experimental group, the CIH mice. These mice were hypoxic. And what we noticed with these mice is that there was a difference in the breathing amplitude in these mice when comparing licking to non-licking. It is significantly lower, but it returns to baseline when they are done licking or close to baseline. This is also reflected in our data. These mice, we noted that they spent less time licking than not licking, and there is a significant difference between licking and non-licking in the bout duration. And there's also a significant difference in the root mean square amplitude as well. And this bottom graph here, we decided to look at the lick bout duration between these two groups and found that even though it looks like there might be a difference, there actually is no statistical change between these two. In addition to looking at coordination between breathing and licking, we wanted to look at the sex differences between these groups as well. And we actually noted that there were no differences between these groups, but we also looked at the coefficient of variation and found that males had a higher variance in lick bout duration than the females did.
And then for limitations, like I said, because of how messy it could get because of external factors, we ran an outlier test on our root mean square amplitude calculations to account for all of that could have disrupted our data. And what that kind of looks like, just to give you a visual image, is what is on the bottom, a messy trace versus a clean trace. And secondly, for trial and error, we actually ran this experiment twice. In the first set, it was our first time, so we were still trying to get used to blocking out as much noise as we could. And then the second time, we perfected it a bit more. So you can see that the traces would have improved, and that could have played a factor in our data. So just letting you guys know about that. And then as for sample size, we used a total of 15 mice, which is not relatively small for mice, but could always be better in terms of acquiring data. And then for interpretation, we found that chronic intermittent hypoxia not only disrupts swallow breathing coordination, but also drinking/licking breathing coordination, which is really fascinating because breathing as a whole field has not been studied as much. So to try to measure that via licking, and then on top of that using chronic intermittent hypoxia, is extremely cool in like our opinion because we like geek about this, but also it raises a lot of discussion questions in the Ramirez lab, such as if what really accounts for that female/male data set variability, if there is a specific developmental period in which this apnea effect worsens, and more. So just, yeah, keep that in mind.
And there are a few other things that we actually wanted to share today. There were the things that we learned in the lab and also other projects that we worked on. And one of the things we wanted to share was that we learned how to surgically implant a trach tube into live mice under the guidance of Dr. Huff. This is a schematic that shows where we would place a trach tube, as well as muscles that we record from using electrodes. The second project we worked on was tailored by Dr. Bush. I do want to mention that this was done before, not exactly the same. We looked at if mice liked a high oxygen or normal oxygen environment compared to a low oxygen environment. So you can see that normal oxygen would be 21% and low oxygen would be 10%. And the bottom, you can see this is our data. We're not really going to jump into that given time constraints. The last thing I wanted to share was that we worked on a head fixing project. This was developed by Dr. Bush as well. It is a pilot experiment. He's still working a bit on it, but we wanted to look at recording neurons in the forebrain of mice that could possibly be related to breathing, as this is not something that has been studied or found at all. We invite anybody that is curious about these projects to ask us questions at the end after all the students have presented.
And then these are our references and our acknowledgements. So we'd like to especially thank Dr. Ellenbogen, Mrs. Ellenbogen, Dr. McDonald, Mr. Pridgeon, Julie, and Sylvia, because you guys are awesome. And everyone in the Ramirez lab, 3 of them are here. Hello. We have we have Dr. Huff, Dr. Bush, and Dr. Glover. Yeah, and then there's more listed above. So yeah, thank you so much.